It's a strange thing to think about. A virus isn't really alive, not in the way a bug or a plant is. It can't eat. It can't grow. It can't make its own energy. On its own, a virus is just a tiny package. It's a bit of genetic code, either DNA or RNA, wrapped in a protein shell. That's it. It's basically a microscopic thumb drive with a single destructive program on it. So how does something so simple, so inert, cause so much trouble? How does it bring giants like us to our knees? The answer is that a virus is the ultimate con artist. It's a master of cellular hijacking. Well, it doesn't need its own factory because it's an expert at breaking into ours and forcing our own machinery to work for it. Its entire existence is based on one principle: exploit the living to make more of itself. The process is both brutally simple and incredibly clever. It's a heist executed with biochemical precision. Let's walk through how this heist unfolds step by step. First, the virus has to find the right target. It can't just infect any cell it bumps into. Our bodies are made of trillions of cells, but most of them are the wrong kind. A flu virus that bumps into a skin cell won't do anything. It's looking for something specific. It's looking for a cell with the right door and the right lock on that door. This is the key to the whole operation. The surface of a virus is studded with proteins. Think of them as keys. The surface of our cells is covered in proteins too, called receptors. These are the locks. Our cell receptors aren't there for viruses, of course. They have important jobs. They receive signals from other parts of the body, or they help the cell stick to its neighbors. They are the cell's communication and docking ports. The virus has evolved its protein keys to fit perfectly into some of these cellular locks. This lock and key mechanism is incredibly specific. For example, the SARS-CoV-2 virus, the one that causes COVID-19, has a prominent spike protein on its surface. This spike protein is the key. It's perfectly shaped to fit into a receptor on our cells called ACE2. Cells that have a lot of ACE2 receptors, like those in our lungs, are prime targets. The virus floats around until one of its keys randomly bumps into the right lock. When it does, it sticks. Once it's latched on, the virus needs to get inside. This is the break-in. There are a couple of ways this can happen. One common method is for the virus to trick the cell into inviting it in. Our cells have a process for bringing in nutrients and other useful materials from the outside. It's called endocytosis. The cell membrane dimples inward, wraps around the particle, and pulls it inside in a little bubble called a vesicle. A virus latched onto a receptor can trigger this process. The cell thinks it's engulfing something useful. But it's a Trojan horse. The burglar has just been willingly carried past the security gates. Another way in is more direct. Some viruses, especially those with an oily outer layer called an envelope, can fuse directly with our cell's membrane. Their envelope is actually stolen from a previous host cell, so it's made of the same stuff as our own cell membranes. When the virus gets close enough, the two membranes merge, like two soap bubbles, becoming one. The contents of the virus are then dumped directly into the cell's cytoplasm, the jelly-like substance that fills the cell. This is a fast, efficient entry. It's like melting through the wall instead of picking the lock. So the virus is inside, but it's still in its protective packaging. The genetic code is locked inside its protein shell called a capsid. The next step is uncoding. The virus has to get its malicious code out. The cell's internal environment starts to break down the capsid. The enzymes inside the cell, or the acidic conditions inside that vesicle it was brought in on, dissolve the protein shell. The viral genes are now free, floating in the cytoplasm of our cell. The heist has now moved from a simple break-in to a full-blown takeover of the control room. This is where the true genius of the virus shows itself. Its goal is to get our cell to read its genetic blueprint and start manufacturing new viruses. How it does this depends on what kind of blueprint it carries. Let's start with DNA viruses like herpes or smallpox. Their genetic code is written in DNA, the same language as our own cells' master blueprints. Our cells keep their own DNA safe inside a central compartment called the nucleus. The nucleus is the cell's headquarters. It's where our DNA is read and copied, so the viral DNA often makes its way to the nucleus. Once inside the nucleus, it has a couple of options. Sometimes the viral DNA just hangs out, and our cell's own machinery mistakes it for our DNA. Our enzymes, called polymerases, which are responsible for reading DNA and making copies, start reading the viral DNA instead. They start transcribing the viral genes into messenger RNA, or mRNA. This mRNA is the working copy of the instructions. In a more sinister move, some DNA viruses can actually cut into our own DNA and splice their genes right into our chromosome. The virus becomes a permanent part of our cell's genetic code. Every time that cell divides, it will copy the viral DNA along with its own. This is a lifelong infection. The virus can lie dormant for years, a hidden piece of code, waiting for the right signal to reactivate and start making new viruses again. This is what the chickenpox virus does when it hides in our nerve cells, only to reemerge decades later as shingles. Now, RNA viruses are a bit different. They are more common and include viruses like influenza, the common cold, Ebola, and SARS-CoV-2. Their genetic code is RNA. Most of them don't need to go to the nucleus. They can do all their work out in the cytoplasm, where the cell's protein-making factories called ribosomes are located. There are a few types of RNA viruses, and their strategies are clever. Some are called positive-sense RNA viruses. You can think of their RNA as being a ready-to-go message. It's already in the format of mRNA. As soon as it's released into the cytoplasm, the cell's ribosomes can latch onto it and start reading it directly. The ribosome doesn't know it's reading a foreign blueprint. It just sees a strip of RNA with instructions, and it follows them. It immediately starts building viral proteins. It's the most direct and efficient strategy. The virus gets its proteins made almost instantly upon entry. Then there are negative-sense RNA viruses like measles and rabies. Their RNA is like a photographic negative. Our ribosomes can't read it directly. It has to be converted into a positive-sense copy first. To do this, the virus has to bring its own special tool. It carries its own enzyme called an RNA-dependent RNA polymerase packed right inside its shell. When the virus uncoats, this enzyme gets to work, making positive-sense copies of the viral RNA. These new copies can then be read by our ribosomes to make viral proteins. It's an extra step, but it works. But perhaps the most cunning of all are the retroviruses. HIV is the most famous example. A retrovirus is an RNA virus, but it has an almost unbelievable trick up its sleeve. It wants to become a permanent part of the host cell's DNA, like a DNA virus, but its code is in the wrong language. It's in RNA, so it performs a forbidden act of molecular biology. It does something our cells never do. It reverse engineers its RNA code back into DNA. It carries a special enzyme to do this, called reverse transcriptase. This enzyme reads the viral RNA and uses it as a template to build a matching strand of DNA. It turns the RNA message back into a DNA master blueprint. This newly made viral DNA then travels to the cell's nucleus, and with the help of another viral enzyme gets inserted directly into our own genome. Just like that, the takeover is complete and permanent. The cell is now forever compromised. From that point on, it will carry the instructions to make HIV. It will pass those instructions on to all of its daughter cells. There is no getting rid of it, regardless of the strategy: DNA, positive-sense RNA, negative-sense RNA, or retrovirus. The end goal is the same. The viral genes have successfully commandeered the cell. Now the production phase begins. Our cell, which should be busy making proteins to repair itself, communicate with other cells, or carry out its normal functions, has become a zombie. It is now a dedicated virus factory. The cell's ribosomes are churning out viral proteins. What are these proteins? Some are structural proteins. These are the building blocks for the new viruses, the little pieces that will form the capsid shells. Others are functional proteins. These are enzymes that the virus needs for its own purposes. For example, the virus will make its own polymerase enzymes to make thousands of copies of its genetic material, whether it's DNA or RNA. It will also make enzymes called proteases, which act like molecular scissors, chopping up long chains of viral proteins into smaller functional parts. At the same time, the virus is making countless copies of its own genome. The cell's resources, its energy, its building blocks like nucleotides and amino acids are all being diverted to this single task. The virus is a terrible tenant. It doesn't just use the apartment, it rips out the plumbing and wiring to build its own stuff. Many viruses are even more aggressive. They don't just use the cell's machinery, they actively sabotage the cell's own operation. They might produce proteins that chop up the cell's own mRNA, so our ribosomes can only read the viral messages. Or they might block the signals that would tell the immune system that the cell has been invaded. They shut down the host systems to ensure all resources are dedicated to their own replication. It's a complete and hostile corporate takeover. The original management has been locked in the closet, and the raiders are running the company into the ground to liquidate its assets. So now the cell is filled with all the necessary components. There are thousands of copies of the viral genome and thousands of protein pieces for the new capsids. The next step is assembly. This part is almost magical in its efficiency. The new viruses basically build themselves. The structural proteins are shaped in such a way that they naturally attract each other and fit together like a sophisticated form of Lego. They spontaneously assemble around a copy of the viral genome, forming a new, complete capsid. This process, called self-assembly, requires no extra energy or instructions. It's a marvel of biochemical engineering. The parts are designed to click together perfectly. Now, we have thousands of new, fully formed viruses packed inside the dying host cell. The final step of the heist is the getaway. They have to get out to go and infect other cells. Again, there are two main strategies for this. The first is brutal and effective lysis. The virus produces enzymes that break down the cell's own membrane from the inside. The cell literally bursts open. This explosion, or lysis, releases all the new viruses at once in a massive wave. This, of course, kills the host cell. It's a one-and-done deal. The factory is destroyed in the process of shipping the product. Many common viruses, like the one that causes the cold, use this method. The sore throat and runny nose you get are partly the result of millions of your cells bursting open. The second method is a bit more subtle and in some ways more insidious. It's called budding. This is used by enveloped viruses like influenza and HIV. The newly assembled viruses move to the edge of the cell. As they push their way out, they wrap themselves in a small piece of the host cell's outer membrane. This stolen membrane becomes their own envelope. This strategy has two huge advantages. First, it doesn't necessarily kill the cell right away. The cell can survive for a while, continuing to pump out new viruses. It becomes a long-term factory. Second, the stolen envelope acts as a disguise. The new virus is cloaked in a membrane that looks just like our own cells. This can help it evade the immune system, at least for a little while. It's like the burglars leaving the building wearing the security guards' uniforms. And that's the cycle. One virus enters one cell. A few hours or days later, thousands of new viruses emerge, ready to go and repeat the process in neighboring cells. The infection spreads exponentially. One becomes 1,000 become a million become a billion. This is why you can feel fine in the morning and terrible by evening. The viral load in your body is skyrocketing. So why is this process so brutally effective? There are a few key reasons. First is its simplicity. The virus travels light. It carries only the bare minimum genetic information it needs. It doesn't waste energy on anything it can steal from its host. Our cells are complex, with thousands of genes and intricate systems for energy production, repair, and regulation. The virus ignores all that. It just needs the parts that read genes and build proteins. Its minimalism makes it nimble and efficient. Second is its speed. The viral replication cycle can be incredibly fast. Some viruses can go from entry to exit in a matter of hours. This rapid multiplication can overwhelm the immune system before it has a chance to mount a full response. By the time your body realizes it's under attack, there are already millions of viral particles spreading. Third is its complete reliance on our own systems. This is the core of the con. The virus uses the very machinery of life against us. The tools our cells use to stay alive, our polymerases, our ribosomes, our energy sources, are the same tools the virus uses to destroy us. This makes it very difficult to fight a virus with drugs. A drug that blocks protein production to stop a virus would also block our own cells from making proteins, which would kill us. That's why antiviral drugs have to be so specific. They need to target a process that is unique to the virus, like the reverse transcriptase enzyme in HIV, or the protease enzyme that chops up viral proteins. But this isn't a one-sided story. Our bodies are not passive victims. We have been in a war with viruses for millions of years, and we have developed sophisticated defense systems. This is the great arms race of biology. Our cells have their own internal alarm systems. When a cell detects it's been infected, it can send out distress signals. It releases proteins called interferons. These interferons act as a warning to neighboring cells. They tell them, "I'm infected. Lock your doors. Prepare for an attack." The neighboring cells respond by beefing up their antiviral defenses, making it harder for the virus to get in and replicate. Sometimes a cell knows the fight is lost. To protect the rest of the body, it can commit suicide. This process is called apoptosis, or programmed cell death. The cell systematically dismantles itself, neatly packaging up its contents so it doesn't cause a mess and trigger inflammation. By killing itself, it destroys the virus factory before the new viruses can be released. It's a heroic sacrifice for the greater good of the organism. Then there's our immune system, a powerful and specialized army. We have cells like T cells that patrol our bodies, checking the identification of every cell they meet. Our cells constantly display little fragments of the proteins they are making on their surface. A patrolling T cell can read these fragments. If it finds a cell displaying viral protein fragments, it knows that cell has been hijacked. The T cell will then order the infected cell to commit suicide, or it will kill it directly. We also have B cells, which produce antibodies. Antibodies are like smart bombs. They are Y-shaped proteins that are specifically designed to recognize and stick to the proteins on the outside of a virus. When antibodies code a virus, they can neutralize it. They can block its keys so it can't attach to our cells. They can also tag the virus for destruction, flagging it down for bigger immune cells to come and gobble it up. This is what a vaccine does. It shows our immune system a piece of the virus, like its spike protein, without causing an actual infection. Our B cells and T cells learn to recognize this piece. They create a memory of it. So if the real virus ever shows up, our immune system is already prepared. It can produce the right antibodies and T cells immediately, neutralizing the virus before it can get a foothold. But viruses are not static targets. They are masters of evolution. Because they replicate so quickly and sloppily, their genetic code is constantly making mistakes or mutations. Most of these mutations are useless or harmful to the virus. But every now and then, a mutation gives the virus an advantage. Maybe a mutation changes the shape of its surface proteins just enough that our existing antibodies can no longer recognize it. This is called antigenic drift. It's why we need a new flu shot every year. The influenza virus is constantly changing its code, forcing our immune system to play catch-up. Sometimes even more dramatic changes happen. If two different flu viruses infect the same cell at the same time, they can swap entire segments of their RNA. This is called antigenic shift. It can create a brand new virus that is completely unfamiliar to our immune system, which can lead to a pandemic. Viruses have also evolved ways to fight back directly against our defenses. Some can block the interferon signals. Others can produce proteins that prevent the infected cell from displaying viral fragments on its surface, making it invisible to T cells. And some, like HIV, take the most direct approach possible. They specifically attack and destroy our immune cells, dismantling our body's army from the inside out. Other viruses, like herpes, play the long game. They have a strategy called latency. After the initial infection, the virus doesn't get completely cleared. Instead, it goes into hiding inside long-lived cells, like our neurons. It shuts down almost all of its genes and goes dormant. In this state, it produces very few viral proteins, so there's nothing for the immune system to see. It just sits there, a silent piece of DNA in our cells, waiting. Years later, if we get stressed or our immune system is weakened, the virus can reactivate and cause another outbreak. So, the hijacking of our cells is effective because it is a strategy honed by billions of years of evolution. The virus is a minimalist parasite. It strips down the problem of existence to its absolute core, make more copies. It outsources everything else. It doesn't need to find food or breathe or maintain its own body. It lets our cells do all the hard work. It simply provides the blueprint for its own replication and turns our own life-sustaining machinery into an engine of its spread. It is a story of keys and locks, of Trojan horses and secret messages. It's about sabotage, deception, and a relentless, mindless drive to replicate. It's not evil, it's just biology. And understanding the intricate, elegant, and terrifyingly effective ways a virus hijacks our cells is the first step in learning how to fight back. It's a constant, microscopic war being waged inside us, and the virus is a formidable and often successful opponent. Its success is a humbling reminder that in nature, complexity and size are no guarantee of dominance. Sometimes, the simplest plan, executed perfectly, is the one that wins.